Greetings to all the viewers. Now we are going to discuss about the alkaloid called nicotine. Nicotine is the natural alkaloid which is extracted from the tobacco leaves, and the session whatever we are going to cover is AI powered. So let's enter into the session whatever we are going to discuss about. Nicotine is an alkaloid found in tobacco plants. So Nicotiana tobacum is the scientific name of that compound, and it belongs to the class of compounds called alkaloids. Nicotine is the part of alkaloid. Nitrogen is the essential organism present in that. So this is the chemical composition of that nicotine, and it is the part of alkaloid. Which are naturally occurring organic compounds, mostly contains nitrogen atoms and pronounced physiological effect on humans and animals. They are able to work upon the human as well as on the animals, so that they are potential physiological components. Nicotine is known for its stimulant effect. So whenever nicotine consumed, it affects on the nervous system, so that it works like a stimulating, activating agent it will be. So stimulant for the nervous system and is the addictive compound in tobacco product. So whenever continuously used this product, it is having addiction property. So still the people are able to tend to get that one. That is the way addiction property is the common side effect for this one. Apart from that, some useful medication effects also we can notice for this nicotine. First of all, let's see what is chemical structure. Nicotine is consisting pyridine ring and pyrrolidine ring. So this is the perfect aromatic heterocyclic six-membered ring called pyridine, and this is the saturated five-membered nitrogen containing ring called pyrrolidine, right? So both are directly connected, and nitrogen is accompanied by N-methylation. And here we can depict the stereochemistry as well. Stereochemistry around this carbon also perfectly mentioned. This is the way pyridine, pyrrolidine kind of compound it will be, and nitrogen atoms on both the rings contributes the classification of alkaloid. Alkaloid essential component is nitrogen. Here we can notice heterocyclic nitrogen on both the cyclic rings, so that it is the part of alkaloid sources. From where you can extract this nicotine? Tobacco plants. Tobacco plants are the common source for this nicotine. Other plants like tomatoes, potatoes, eggplants, and green pepper contains a traces amount of nicotine. Maximum concentration we can economic amount we can achieve from this tobacco plant. Apart from that, tomato, potato, eggplant, green pepper, in that also nicotine is there in the infinitesimal amount. Biological effects. What kind of effect we can notice in the human organisms? Nicotine works by mimicking neurotransmitter acetylcholine and binding to nicotinic acetylcholine receptors in the brain. So here receptoric site will be there. What is that? Nicotinic acetylcholine receptor, that is called NACHRS. So this is the receptoric site where neurotransmitter is able to bind, so that dopamine will be secreted. The dopamine, whatever secreted, that used to stimulate the activity of the neurons. Neurons are the fundamental particles of the entire nervous system. The neuron activation got carried by means of this dopamine. Dopamine secretion is very essential to get the harmony among all the kind of activities. So whatever response we have to give, that is perfectly controlled by our brain, and it's a signals. That entire signaling system is given by neurons, neurological system it will be, right? So that whatever nicotine, if we are consuming nicotine inside, so whenever nicotine consumed, it also having exact resembling effect of this neurotransmitter acetylcholine. Acetylcholine will bind to this receptoric site, right? So it will replace, acetylcholine will be replaced with this nicotine, so that it able to bind with that brain receptor. Whenever this nicotine is binding upon this acetylcholine receptoric site, it able to release the neurotransmitter such as dopamine. Here also we are able to get dopamine as well. By this nicotine also secretion of dopamine is possible, so that that is able to give the pleasure and the reward, and that kind of a pleasantness we will achieve whenever nicotine consumed over there. But continuous consumption leads to addiction property, right? So dopamine secretion is required in the infinitesimal level only. Whenever nicotine consumed, its concentration got enhanced, so that able to give abnormal condition, abnormal symptoms we can notice over there. And moreover, addiction property is the common thing over this nicotine consumption. And the uses recreational, most of the people for recreation purpose, for joy, they able to consume. And in the cigars and other tobacco products also present, so that is of addictive in nature. And apart from that, we have health benefits as well. Therapeutic usage we can notice. Nicotine replacement therapies, how can we use? Nicotine itself is an addictive drug, then how can we retard that addiction property by using again nicotine only? This is purely explained by means of, in order to minimize the poison tendency of the venom of the snake, another anti-venom also synthesized from the snake only. That means, so whatever poison is generated by the venom of the snake, another anti-venom again it is synthesized from that snake poison only. In the same manner, in order to remove the addiction property of the nicotine, again nicotine itself used as a replacement therapy. Nicotine replacement addiction can be slowly reduced. That is the way we can use. And another example I would like to mention over antibiotics to kill the pathogens, right? That might be a bacteria or virus, but they also again synthesized from another microorganisms only. They extracted from microorganisms and they used to kill another microorganism. That's the reason why here nicotine also having the replacement therapy. They used to remove or destroy the addiction property, those who are addicted for the smoking process. So this is the way therapeutic usage you can notice. Structural elucidation of nicotine involves determining its molecular structure, including arrangement of atoms and functional groups. How the atoms are arranged and what will be the arrangement or localization of their functional groups, we are going to determine in the systematical manner under the segment called structural elucidation. According to structural elucidation process by analytical methods, molecular formula, elemental analysis we are going to do. Elemental analysis means individual elements whoever present in this nicotine can be identified. The elements present in the nicotine are carbon, hydrogen, nitrogen, and their contribution will be C10, H14, and N2. This is the molecular formula which is calculated by elemental analysis process. And here you can notice 6-membered ring. We can find 5 carbons, 6th, 7th, 8th, and 9th, and here another 10th carbon. So that C10 carbons are there, and 2 nitrogens are located on 2 heterocyclic rings, and remaining are contributed by hydrogens only. This is the way molecular formula for the nicotine is C10H14N2. Let's go with another instrumental method called mass spectroscopy. Mass spectroscopy, the important application is molecular weight determination. So here MZ values are calculated. MZ of this nicotine is calculated as 162 gram per mole. Fragmentation pattern. In order to make the convenience for the structural elucidation, we have to strategically fragment the molecule so that individual fragments give the more information in the simpler manner. That's a reason why fragmentation giving rise to the information regarding functional groups located on this nicotine molecule. And we entered into another instrumental method called the infrared spectroscopy. In order to elucidate this nicotine structural composition, here infrared spectroscopy is plotted on wave number in centimeter inverse versus transmittance on the y-axis. You can notice the distinct peaks which are used to characterize NH peak of 3300 centimeter inverse and CH stretching at 3000 to 2850 centimeter inverse and C double bond and stretching 1600 centimeter inverse. These three characteristic peaks we can notice under IR spectroscopy analysis process, so that here nitrogen is there, carbon hydrogen stretching, double carbon double bonded nitrogen stretching we can notice in this what nicotine compound. Here is the systematical representation of curvatures we can observe wherever distinct peaks that corresponds to the these three stretching vibrations. And the nuclear magnetic resonance NMR spectroscopy. Here you can clearly observe the NMR peaks NMR peaks for this nicotine molecule, and nicotine hydrogen environment can be calculated over there. Peaks corresponds to the pyridine ring laying in 7 to 8 ppm range. The shielded protons are studied over, and signals aliphatic hydrogen, where you can locate aliphatic hydrogens on this methyl. These methyl hydrogens are aliphatic in nature, which are giving the peaks at 1 to 3 ppm. So here upfield shift you can notice over on the pyrrolidine ring. This is called pyrrolidine, saturated 5-membered ring pyrrolidine. On that you can notice methyl, whose protons are giving the signal at 1 to 3 ppm. This is regarding the information of nuclear magnetic resonance spectroscopy. Let's dive into the C13. C13 giving rise to the nature of the carbon atoms. Aromatic carbons are giving the chemical shift value around 120 to 150 parts per million. Aliphatic carbons are at a lower value, that is 10 to 70 ppm. This is regarding pyrrolidine carbon atoms. So this is an informative analytical analysis for the carbon environment by means of C13 NMR. Let's go with the UV visible spectroscopy, ultraviolet visible spectroscopy. So within the visible segment, we can notice this spectrum and broad signals. We can notice wavelength versus absorbance was plotted, and you can notice this kind of curvatures in UV visible spectroscopy. The purpose of UV visible to identify the conjugation. Let's see whether conjugation is present within the molecule or not. Obviously, all the aromatic ring double bonds are conjugation. They are in the conjugation. Alternative arrangement is located so that it is active towards UV visible spectroscopy. Hence we can notice the broad spectrum lines for UV visible spectroscopy. X-ray crystallography. X-ray crystallography is meant for the crystal nature and bond lengths and the fixation of the atoms in the particular rigid three-dimensional domain of that particular molecule. If the crystal of nicotine is one of its derivatives available, X-ray diffraction can give exact 3D structure. So X-ray diffraction is used to give the nicotine 3D structure, conforming the bond lengths and angles, spatial arrangement of the atoms. All information you can achieve by means of X-ray crystallography technique. Let's go with the summary. So let's summarize the entire nicotine structural elucidation by instrumental method. Here shaded regions are provided for the information regarding the individual fragments of this nicotine. Pyridine ring. So here whatever blue shade you can observe, that is aromatic 6-membered ring system with the nitrogen as the heteroatom, and this is responsible for the basic nature. Pyridine is basic in nature, everybody aware about that one. Though it is in conjugation, it is the part of aromatic ring, but the electrons are free for the donation. They are residing outside the nitrogen, residing outside the aromatic sphere, so that basic nature is imparted for the molecule by means of pyridine ring. Pyrrolidine. So another 5-membered ring of saturation is regarded as a pyrrolidine ring. It is of 5-membered with the nitrogen atom and attached to the pyridine through the carbon-carbon linkage. Here carbon-carbon linkage, so that both the heterocyclic rings got combined. Alkyl chain. Upon nitrogen, we can observe the methyl. This is regarded as a tertiary amine. So in alkyl side chain is located over there. Pyrrolidine ring, alkyl substituted contributing volatility and bioavailability. The purpose of methyl group over here that we have multiple applications for this methyl. It is the compound nicotine whenever absorbed that easily inhaled inside. That is because of this methyl. Volatility is imparted by means of this methyl and bioavailability. So whatever biological impact we notice, that is also responsible by this methyl. These techniques together conforms nicotine as a bicyclic compound. So here two ring systems you can notice. One is of pyridine, another is of saturated pyrrole ring, right? So it is the biannular system, bicyclic system it will be. And now let's go with the synthetic process. How can we synthesize the nicotine? So first let's go with the natural biosynthetic process, naturally extracted from the nature, and alternatively we will go with the parallel synthesis in the laboratory, right? The synthesis of nicotine can be performed through the various methods, both biosynthetically in the nature and artificially in the laboratory. Both the methods, from the nature you can extract them, and resembling substance also we can synthesize in the laboratory by means of the process called research. Below is an overview for the natural biosynthetic path and laboratory chemical synthesis for this nicotine as well. Both the methods we are going to discuss in detail. Natural biosynthesis of nicotine. Nicotine is primarily synthesized from the roots of tobacco plants. Nicotiana tobacum is the scientific name of the compound. In the step-by-step process, first of all, we have to take two precursor compounds, starting materials are two in a biosynthesis process. One is called nicotinic acid. It is a derivative of pyridine. Here you can clearly observe the nicotinic acid. Pyridine 3-carboxylic acid is called nicotinic acid, and here we mentioned nicotinamide. This is not required actually. Nicotinic acid we are thinking about N-methyl pyrrolinium cation. N-methyl pyrrolinium cation is nothing but that is the second fragment. Whatever second part we have, this is another fragment. So this is the first fragment with the carboxylic acid over, and this is the second fragment we have to use. These two are taken as the starting materials in order to combine. Steps in the biosynthesis. Synthesis of nicotinic acid. How can we synthesize this nicotinic acid? Aspartate is converted into quinolinic acid via the series of reactions. Quinolinic acid is cyclized into nicotinic acid, pyridine ring precursor. So this is the way aspartate is the starting material which further transformed into quinolinic acid. That quinolinic acid, that quinolinic acid can be cyclized, giving rise to the nicotinic acid, which is of pyridine ring. This is the way synthesis of nicotinic acid get carried. Let's go with the steps involved in the biosynthesis. Nicotinic acid. We taken, and this nicotinic acid pyridine ring precursor was taken, and the formation of N-methyl pyrrolinium cation, ornithine cycle will be there, and an amino acid undergoes a decarboxylation to form putrescine. Putrescine undergoes methylation and cyclization to form N-methyl pyrrolinium cation. Nicotinic acid and N-methyl pyrrolinium cation undergo pictet Spengler-like condensation to form nicotine. This is the way extraction process got carried. First of all, we have to take nicotinic acid. That nicotinic acid was taken, and ornithine is another material taken. This ornithine is a amino acid. Ornithine is a kind of amino acid. Amino acid means which consisting in H2 and COOH. So this ornithine is decarboxylated. Whenever decarboxylated, only amino part will be present over there. After the removal of this carbon dioxide, whatever amino compound generated from this ornithine is called petricin. That petricin further methylated. Whenever it is methylated, N-methyl pyrrolinium cation will be generated. Whatever this fragment, orange color shade material you can notice, this part will be synthesized from the ornithine. After getting this ornithine, already we have nicotinic acid. So that nicotinic acid and this pyrrolidine compound both are subjected to a named reaction called Pictet-Spengler-like reaction. Pictet-Spengler type of condensation reaction got carried, so that we are able to achieve nicotine as a final product. By this, the synthetic route, the biosynthetic pathway, which is proceeded with the nicotinic acid and N-methyl pyrrolinium cation as the precursors in order to synthesize the final product nicotine. Now we entered into another segment called laboratory method. So all these alkaloids, whatever extracted from the plants, we are searching for alternative synthesis in the laboratory as well. So that lab synthesis we will go with by taking 3-pyridyl aldehyde as a starting material. So this 3-pyridyl aldehyde taken as a starting material further subjected to Mannich reaction, and cyclization got carried, further reduction, and final product will be achieved. These many 5 steps we can notice that will be categorized, that will be presented in the form of schematic representation over there. Starting, we have to take what his compound name is nicotinic acid, synthesized from 3-pyridyl aldehyde. This is called 3-pyridyl aldehyde, treated with tosylamine. This is called tosylamine. Tosylamine, when treated over, it will be condensed. So this water, removal of water giving rise to a double bond NTS, which is called water condensed product. Condensed product, further Grignard reagent will be added. This is called a Grignard reagent. So this Grignard reagent will be added at this active carbon atom. So that active carbon atom will be this active carbon atom will be attached to this carbon. Obviously, here carbene intermediate will be generated. Being magnesium is a electropositive metal, so that here carbon is resided with the negative charge. Negative charge carbon, whenever moving over here, the double bond is displaced towards the nitrogen atom, and here we can observe the side chain with the ether linkage, because we are taking the starting material with the ether linkage. No, that's the reason why ether side chain will be appeared. Further, we have to go for the iodine, addition of iodine over there, and CH2 dichloroethane is also added. Phenyl iodo aryl carboxylate is also added. All these reagents are subjected to this compound, giving rise to a cyclization process. Cyclization not carried with this oxygen, rather already if we aware that ether is a chemically inert functional group. That's the reason why the adjacent carbon will participate in the cyclization reaction, giving 5-membered ring. Is 5-membered ring with the side chain ether linkage will be there, and we are subjecting this product whatever appeared with the sulfuric acid and heated at 80 degrees centigrade. Now we have to go with the reduction with sodium borohydride. By means of that reaction, we can achieve one pyridine ring. Already you can notice even in the starting material as well, and the side chain, whatever the side chains are present on this pyrrolidine, entirely removed. We got the saturated pyrrolidine ring over there. This saturated pyrrolidine ring now treated for formic acid, and also what it is HCHO, that is called formaldehyde. At 80 degrees centigrade, the reaction giving rise to N-methylation. N-methylation or N-alkylation, we can denote it as this is the entire route map for the nicotine synthesis. started with the pyridine aldehyde compound and the systematic approach in order to accord this final product nicotine. So all the reagents are very essential in the quantified molar ratio, so that their molar ratio is also perfectly given and percentage of yield is also mentioned for each and every step. This is the entire statistical representation of that chemical synthesis of nicotine within laboratory. After synthesizing the nicotine within the laboratory, and the next segment we are going to discuss about the applications where we can apply this nicotine. Though it is having addiction property, any material in the entire universe is never treated it as a waste one, because whatever the material you find, everything will have its own way of applications. The only the thing we have to know is in what way we have to use it, in what way we best use it. That is only the thing we need to understand. Then only each and every product will be important. Though it is having addiction property, though it is extracted from the tobacco, though it is having the constraints in the medical consumption, but systematic usage may cause the applications in the medical fields as well. Nicotine has a wide range of applications, although it is most commonly associated with tobacco products. Its effects in the nervous system as well as in the chemical properties, giving rise to the applications in the various fields. So many applications you can notice because of its distinct properties. Even in the medicine, agricultural fields, research, everywhere we can use nicotine as an um, effective usable material, effective chemical component. Here, medical application, nicotine replacement therapy, as we already discussed about that one. This nicotine replacement therapy means those who are already addicted completely for that nicotine or else tobacco products. Now we can use nicotine replacement therapy, which is called NRT, and those are available in the form of patches, gums, lozenges, and sprays, inhalers as well. So whenever we are consuming them, the people slowly de-addicted towards that nicotine, slowly coming out from the sphere of that nicotine addiction property. So these are called what, nicotine replacing therapy. And they also made by means of this nicotine as the ingredient, that is the medicinal material used in a de-addictive chemicals. These therapies reduce the withdrawal symptoms, cravings, and helping smokers to gradually reduce dependence on the nicotine. Whenever such materials, such chemical components are consumed in the requisite amount, the addiction property is possible for these smokers. Potential treatment for neurodegenerative diseases, neurological systems, the millions of neurons are working effectively with the coordination among them. Entire system is working. Whatever things we are seeing, immediately we will respond to that one. That is purely depends upon the nervous system only, right? So neurodegeneration is nothing but that kind of coordination will be missing, and whatever information reached to the brain, and that response which is coming to the external region, the entire system will be disordered, which is called neurodegeneration. Neurodegeneration means destroy of the nervous system proper functioning. Because of that reason, Alzheimer's, we are not able to remember all the things in a proper way, that is called Alzheimer's. Research suggests that nicotine may have protective effects against cognitive decline of Alzheimer's. Optimal consumption reduce the Alzheimer's disease, Parkinson's. Being nicotine is a kind of neurotransmitter, can effectively binds to the brain receptoric site, so that it will secrete the dopamine. So it will be the alternate for the natural acetylcholine, and the Parkinson disease will be minimized by the consumption of nicotine in the prescribed dosage form. Cognitive enhancement. Nicotine has been studied for the potential improving the attention, memory, and cognitive performance as well. And it stimulates the central nervous system. being it is purely working upon the nervous system functioning, so that nicotine consumption in the particular level, particular dosage form, it effectively improves the working tendency of the central nervous system, weight control. It also what consumption of nicotine also controls the weight. The important applications of nicotine in the agricultural field, being nicotine is a kind of a very strong chemical. We can use its toxicity into a usable form by making insecticides. Nicotine sulfate is available in the market as a commercial pesticide, which able to remove the pests like aphids and caterpillars. And so they are used to sprayed on the cropland, so in order to mitigate pests which are damaging the croplands naturally. So this is the way organic farming included with this insecticide nicotine, precursor for the synthetic insecticide. Though we are not directly using this nicotine as a starting material or else a product for the pesticide, we can use it as a starting material to synthesize further effective pesticides also. So that starting material, it works like a starting material to synthesize effective pesticides also. This is the agricultural applications found for that one. Scientific research. Neuroscience. Purely nicotine is related with the brain functioning and brain stimulation property. So that neuroscience and how, what extent and how frequent the people are consuming, and addiction property is possible, and what is the dosage, what is the condition, what is the time duration. Entire thing got studied by this scientific research under this category called neuroscience. Nicotine is widely used in the research to study the mechanism of addiction, why they are addicting to that one, why that kind of addiction tendency is there. The study was carried. Its interaction with the nicotinic acetylcholine receptors is of great interest to understand addiction develops, and how it can be treated, how it is binding to the receptor, and how can we de-addict it to that when the entire research is going on. Cognitive function research. Nicotine is also used to study investigating learning, memory, cognitive enhancement. How consumption of nicotine will impact our memory, learning process, and cognitive enhancement. All these get under observation. Drug development. Scientists use nicotine as a lead compound to develop further drugs. Just now we discussed, being it is a kind of effective pesticide, effective medical applications containing material, so that we can use nicotine as a starting material to synthesize further effective medication. Tobacco products. Tobacco products, nicotine is the primary active ingredients in the cigars, and smoking tobaccos and e-cigars will be there. All these are composed by this kind of nicotine. E-cigarettes also present. E-cigars without any risk, the people can consume. Industrial and biochemical applications, biosynthetic studies. Nicotine serves as a model compound in the biosynthesis of the plants. It has been used in research to understand plants to produce secondary metabolites, and this leads to the agricultural practices in a better way, and bioengineering is the output of this entire process, right? So biosynthetic studies are crucially studied under the category called bioengineering. Phytochemical extraction, whatever crude product we are extracting from the plant, which is called phytochemical extraction. The extraction and isolation of nicotine from the plant, interest in the phytochemistry, where it is studied for the chemical properties as well as development of environment friendly pesticides. How can we synthesize environment friendly pesticides by using these? So extraction, plant extracts. This study is also another application in industrial and biochemical field. Next, recreational use. Just now we saw that whenever the people are consuming, they are having the feeling of relaxation, feeling of pleasantness. That is the cause for this addiction property. Alternative therapies. smoking cessation devices. New alternative nicotine devices such as heated tobacco products will be there being developed to reduce the harm caused by smoking methods. These products aims to minimize the exposure harmful byproducts. So they used to hate wherever nicotine is there, that repels. So repelling tendency will be there. That is smoking cessation devices are valuable whenever we are under this treatment. They will repel the nicotine, and the slowly de-addiction process is possible. So we are able to conclude the entire session. The compound called nicotine, which is the class of alkaloid, pyridine pyrrolidine type of chemical composition skeleton, it is incorporated with the entire study included with what is the structure, what will be the structural elucidation process, biosynthetic route map, and how can we synthesize within the laboratory. Later on applications for nicotine in the various fields, the study is taken in the detailed manner. Hopefully the session is very informative and it is AI powered session. Thank you for your patient listening. This is Dr. Kavita Kottireddy Kota. Thank you very much.